Morning, morning, morning. How many have we got already? Loads of you waiting. That's nice. Okay, so I'm just actually getting set up a little bit here, but okay, so what I need to just do is mute myself here so you can't hear my echoey feedback. Okay, that's better. Let's mute that. Okay, so I'm just going to screen share. We're going to change the schedule a little bit. In fact, I'll just go through what we're doing today at the end of this week. So we looked at some specific immune response yesterday. We've had a little bit of phagocytosis, so non-specific as well. We've got some primary secondary response to the differences there, which is actually quite relevant with the COVID-19. And we've also got some antibodies and vaccines. So I'm just going to be sort of dipping into some different bits and pieces like that. I can also maybe do some questions. I'm going to be responding to what you guys are will find most useful. I think primary and secondary, given that it is sort of highly relevant right now, I think it's going to be the best thing to do. I haven't sent the documents to myself on this iPad yet, so I think I'll do that first. Let's rearrange some windows here so that I can see everything. Okay, so anything that you've, any questions from anything work we've done so far? Also, your year 12, but it's going to influence the year 13s more, but we have an announcement this afternoon at 4 o'clock. I'm going to be doing a YouTube live about how the calculated grades will be being calculated. So we have some more information that we can share with you later on. So make sure you follow along for that. There will be some information about, we did some Instagram polls yesterday asking you what you are most concerned about. So we'll be addressing some of those as well. So we're also going to be doing our regular Monday check-ins again from Monday. So we'll be there to answer your questions in the evenings on YouTube live, as well as these regular sort of live classes are going to be continuing inside the study at home exambulance. Okay, so we'll be sending all our students an email out this afternoon before 4 o'clock, just to give you that notice about the YouTube live with the announcements later today. Okay, try and get everything. We did a bit about antibodies yesterday, so maybe I can go into the detail of an antibody a little bit more, as well as the sort of the differences between primary response. What is primary response? What is primary immune response? That's something I would like you to answer for me. Okay. Okay, so the defense barriers. Okay, so it's a bit different. The skin being a barrier to infection is different to the primary immune response. The primary immune response is the immune response that we undergo after first infection, primary first infection to a pathogen. So this is the response that we elicit when we do vaccination and before the memory cells are formed, correct. So this is why why COVID-19 is such an issue, is because it's a new pathogen, it's a new antigen. Nobody in the world has immunity to it because it's novel. And therefore, everybody is going through primary immune response, which is when you feel the symptoms. Everyone is basically suffering from this. So then how's that, broadly speaking, different to the secondary immune response? Well, that's when you have previously been exposed to that infection or to a vaccine. And then the vaccine is designed to make you go through primary immune response, or to make those memory cells, but without making you suffer from the disease, whatever that pathogen may cause. So if you've already got the memory cells, then we can say the secondary response produces more antibodies, and it produces antibodies faster, and so that you basically can crush or quell that infection before you feel symptoms. Yeah, it's faster and stronger than the primary immune response. Okay, so I think we can probably do some notes on this, and then I think I might move straight into questions. That's probably, I think, what you guys are most likely to want to do on this. So I'll share my iPad and get some notes. Give me a yes or no to have you covered primary, secondary immune response in school or college yet. So just get a yes or no to that. What can you see the top of my iPad there? I'm wondering. No, just down a shop effect. Okay, so I'll skip over this relatively quickly then, so that we can do more on the question work, which will be probably more valuable to you if you have already covered this once before already. So let's. That is pretty strange. Maybe I'm not going to, maybe we're just going to do questions, because despite having shared my iPad all week, it's telling me that it. Okay, well, we'll see if this works. Interesting. Okay, you should all be now seeing my iPad screen, and I will. Why are you not charging, iPad? Why are you not charging? You can see my iPad is a bit low on juice. Let me just turn the screen brightness down, so it shouldn't affect what you guys see on the front end. I'm just going to have to run on low power mode in the back end. Okay, so let's try that again, shall we? It wasn't in my PDF viewer. Everything's just copy to PDF expert. And that's more like it. Here we go. Now I can write on it. So I'm going to do the primary and secondary immune response first. So in primary response, so it's your first immune response, basically. Maybe let's just zoom in a little bit over here. It's slow. Why is it slow? Because you have to go through the full system. So it's going to go from basically phagocytosis, which is going to present the antigens to activate the T cells, which is going to stimulate the B cells, which are going to make plasma cells, which are going to release all of those antibodies that you're going to use to fight the infection. So during this time, the pathogen is going to multiply and you're going to feel symptoms. And then after the infection, you're going to save the memory cells. So that is sort of the end of the primary response. Why can't I? Generally speaking, we're going to be talking about memory B cells. Memory T cells do exist. I'll put T and B cells, but it's predominantly the B cells. Okay, so on my graph here, I'm going to have, let's do it in red. I'm going to have the feel symptoms line, which is one that I'm basically making up. I'm just going to have that as this is our basically number of pathogens. I wonder if I can squeeze that in down the side here. So this is like the symptoms threshold line in my red. And then in green on this y-axis on the left-hand side, this is going to be our immune response. Here. So with our primary response, what color should I do that in? And no, red. Well, let's do the number of pathogens first. So the pathogens I'm going to draw in red from point of first exposure down here. We're going to end up with an increase. We have primary response. We're going to cross that threshold at which at this point is where we start to feel symptoms. And obviously it goes up and up and up. We feel symptoms, and eventually our immune system gets on top of it. We produce the phagocytosis, then the T cells are activating, the B cells are activating. We're producing all those antibodies, and eventually we're producing enough antibodies in enough numbers to quell the infection. And then basically the number of pathogens goes right down, more or less to zero. I mean, you might have some residual in you, but effectively the number of pathogens. So this is our primary response taking place in the number of pathogens. Our immune response, the red line is only to do with the pathogens. So how does our immune response? Basically, it's going to follow that line more or less. I'm going to do this. Yeah, let's do this line in blue. Our immune, I actually should probably do it in green, seeing it's matching the color of the axes. We have our immune response is going to slowly increase, and it's probably going to come down something slightly afterwards, something like this, and then some period of time. So this is basically our immune response. Some period of time passes. In fact, what have I done wrong with that graph? I have done. Increase, increase, increase. It's going to decrease, but it doesn't go down to zero because we keep those memory cells. So there's still some kind of immune, those memory cells, memory B cells, memory T cells are still going to be in us. And then obviously some time passes, which I'm just going to signify with this wiggly line. And then we get infected by the same pathogen with the same antigens again. And this starts to increase. Now we already have the suitable, the complementary antigens at this time, the antibodies and plasma cells. So this is our secondary response is going to be much stronger. This is a much steeper line, and we're going to produce way more antibodies. And because we're producing more antibodies, then our pathogen infection basically never gets to take hold, and the pathogen infection is wiped out before we get to the feeling symptoms line. So the red is the number of pathogens, basically. And our green is our immune response. I've seen different ways to represent this in terms of how long the secondary response lasts for. Typically, they don't like it to come down too quickly. So I think on this graph, I would draw it something like that. And again, it wouldn't ever get to zero. That would actually strengthen our immune response. If it was going to come down to a level, if I'm going to draw it, I would draw this level being slightly higher than the previous level, because that's why sometimes you'll have booster shots for a vaccine. It kind of depends upon actually the type of vaccine that they're giving and how that vaccine's working. But sometimes upon secondary exposure, then you actually retain, your body's like, oh, actually, we might get this infection again. So it keeps more memory cells. Okay, so that's our basic graph there. What notes can we put on in secondary infection? We can say the same pathogen with the same antigen infects you, because sometimes with influenza, we have the antigen changes regularly. The virus remains the same virus, but the antigens are different, and therefore it doesn't form an antigen antibody complex, and therefore even though the virus is pretty much the same, because the antigen is different, you undergo primary response again. That's why it's very difficult to make a vaccine for flu, because it's changing constantly. It's called antigenic variability. It's not in your spec. So So the same antigen, maybe you're infected by a pathogen with the same antigen. The immune response, or the secondary immune response, is faster. So we've got a steeper line on this graph here. It's stronger, which means our peak here is higher than our peak here. And then the plasma cells produce antibodies which kill the pathogen. It involves the memory B cells and the memory T cells. Predominantly the memory B cells, because they're going to be the ones that are producing those antibodies, but their memory T cells are involved, and you do not feel symptoms. What does it say down here? Plasma cells make antibodies which kill the pathogen. Memory cells can stay in the body for decades. Over time, they do generally decrease unless you're re-exposed to secondary infection, and then they'll be topped up again. It kind of depends upon many factors. We just don't know for COVID-19, for coronavirus. We don't know how long those antibodies are going to last around in the bloodstream. I don't know why my iPad isn't charging. It is connected. I don't have an extension lead. I'm far away from a PowerPoint. I thought my laptop. Okay, I'm just going to come back to you over here. I'll turn my screen off and save that 5%. Okay, so maybe what I can do is find some questions on this, and we can run through some questions together. AQA basically don't ever mention memory T cells. No, they will say memory cells. They don't even give it a B or a T. And typically they'll say the memory cells will produce antibodies, and therefore they're talking about B memory cells. Any questions on that so far before I jump into some questions? So the memory cells, yeah, okay. Well, the memory cells will probably be activated by the memory T cells is probably the process. The plasma cells won't just know on its own. They generally need stimulation. As I said, the immune system is crazily complex. But as far as we're concerned, for the basic understanding, we get infected by the pathogen a second time around. The antigen will form an antigen-antibody complex. And then we're going to be like, oh, okay, now we can basically go into antibody production mode. And then the plasma cells will divide by clonal expansion, just make many copies of monoclonal antibodies, just pump out the antibodies. And the antibodies, you don't need to do any of the selection-y process. We've already got the right template to make the right antibody, and it can just go into antibody production mode. Whereas in the primary response, we've got to do all the work to try and figure out which antibody is the one that we need to make. Cell-mediated response is that done by phagocytes and by cytotoxic T cells, because you're going to be an AQA student if you're asking me that. The humoral response is anything done by antibodies traveling in the bloodstream. So we have done that. It's just I haven't used those terms because OCR and EdXL students don't use cell-mediated and humoral responses as key terms. Difference between memory cells and plasma cells? Well, they basically do the same thing, except plasma cells produce the antibodies during the infection, whereas a memory cell is going to be hanging around in the bloodstream after the infection, and it won't be dividing actively to produce many copies of antibodies. The memory cells are just going to be lingering until secondary infection. What produces, I mean, they do secrete the antibodies because they're secreting them into the blood, but I think they are the ones. Does anybody know? I think the site of antibody production, the protein synthesis that produces the antibody proteins, is in. It can't be in another type of cell as far as I would know about it if it was a different type of cell. They do get funny about that with hormones, especially OCR, about saying cells secrete secreting hormones into the blood. I mean, it depends on the wording of the question. It depends whether the production of the antibody is the important thing for the question, or whether it's the antibody. If the important thing is the antibody acting on the antigen somewhere else in the bloodstream, then it's the fact that they're secreting them which is more important, because that's when they get released from the plasma cell into the bloodstream, and that means the antibody can then travel around independently of the plasma cell. Whereas I don't think it's incorrect to say that the antibodies are made inside the plasma cell, but the plasma cell is not attached to the antibody when the antibody is forming the complex with the antigen. Does that kind of make sense? We can do structure of an antibody. In fact, let me find a structure of an antibody question, and I'll do structure of an antibody, but I'll do it in the context of an exam question, I think. Let me pull these up. Okay, let's make this a little bit bigger. Let's find some old spec stuff so I can share the question without sharing the answer at the same time. Okay, well, I've got a question on primary, secondary that I've come across here, which is quite convenient. So let's do that one first, and I'll find an antibody one next. Okay, so here we've got, let's change the shape and size of this window a little bit. Let's see how that affects what you see on here. Okay, sweet. So figure 4 shows the concentration of antibodies. So this is our basically proportion of immune response in a patient's bloodstream following an influenza vaccination and then subsequent infection with the influenza virus. Okay, so we had the vaccination, we have our small number of antibodies, arbitrary units, which is effectively, well, in fact, we'll come up to the mark scheme in a minute. And then we have the actual infection. Notice how it doesn't go back down to 0. These are our memory cells, we still keep some antibodies in the bloodstream. And then this gradient of the graph here is steeper than this gradient of the graph here. And they don't finish it off. And there's very few questions that do show the end of this line. I guess it would probably varies. So describe where the primary and secondary immune responses are taking place on figure 4 and explain the difference between the two processes. I'll tell you how many marks it is, but I'm going to pause that for you. It is 3 marks. Okay, so Yeah, you feel symptoms because of the pathogen. The pathogen is destroying your host cells, and often your cytotoxic T cells, your T killer cells, are going to also be destroying host tissue. So if you have COVID-19, some of the more serious cases, it's when you end up in a bit like a positive feedback of the immune system, triggering the immune system and releasing all these cell signaling molecules called cytokines, and that amplifies the immune response. And the immune response then basically comes in and destroys lots of tissue, lots of lung tissue. So all the healthy cells as well as the infected cells. Okay, so anyone got an answer to where? So we've got to describe where, primary and secondary. It's 3 marks. So there should be a mark for each of those. I would assume there's a mark for each of those. And explain why. I mean, in my head would make this a 4-mark question. I would go describe where primary is and give an explain point about the primary infection and how it's different, and describe point about where secondary is, and an explain point about where secondary is. That's the safest way to approach this question. So even though it is a 3-marker, in my head, I'm looking for 4 bullet points. Okay, so Louis, primary between the vaccination and the infection, or you could say immediately after the vaccination, the days following the vaccination, probably before the infection is good to add. Secondary, okay, so we've got primary response when after the vaccination, the concentration of antibodies is low at the beginning. Is that a reason? Explain means you have to give a reason. So saying that the number of antibodies is low in the beginning is a description and not a reason. So you've got to really pay attention to those command words to make sure you are doing what the question is asking us to do. Why are there fewer antibodies or slower production of antibodies? Okay, so we said secondaries after the infection. It's a stronger, faster response, but why? We need to know why. Can you get that down to like a couple of words? What's the reason why primary is slow? What's the reason why secondary is fast? Okay, so because memory cells are present, you have a stronger and faster secondary response after infection. Okay, that's really nice summary there. So primary response after vaccination, slower and weaker than secondary response because memory cells, oh no, they already exist. Because maybe say antibodies or complementary antibodies have to be made. You might even just be able to say because primary response is taking place. Obviously, it's a bit of a risk because it's asking us about primary response here. Basically, I'd say that the clonal selection is taking place, if you wanted to say that, because that's again, choosing, finding, sorting out, getting the right antibody that's complementary to the pathogen's antigen takes time. That's effectively why it's slow, and you can't produce them until you know which one you need to copy. So lots of nice answers in there, but again, really drilling into the question, what do I have to do for the question? What do I have to do? And that explain word tells us we have to give the reason why. So we're describing where on the graph it takes place, and then giving a reason as to the differences. Why? And then what is the difference? The difference on the y-axis is the number of antibodies. So we have to then take all of that information and basically say, if I was to rephrase this question, it'd be describe where on the graph primary response is taking place and give reasons why the concentration of antibodies is different to secondary, and then I'd do the same for secondary. So really, really breaking that down. So let's, I'll resume my screen share and come down a little bit so we can see the mark scheme, see how, I'm not always right with these, right? So, yeah, well, we've seen the graph. I'm going to get all the marking guidance in. So primary, just after infection, when the person is vaccinated, And so for one marking point here, we have to state the location of both of them. So we have to state the location of both of them. You don't have to do that in a single sentence, but you do have to say in your answer, primary is after vaccination, secondary after infection. And that's what that and means. You have to give both of them for 1 mark. The primary is slower or delayed because of clonal selection or clonal expansion, or the production of antibodies is slower. So pretty simple answer there. But again, it's looking for that reason. Secondary is faster response, shorter lag time, more antibodies produced. It's really the because of the memory cells or immunological memory. If not stated, look for appropriate labels on the graph, except description. Okay. Any questions about that before I move on? Okay, here's a nice antibody question. I'll answer your questions before. No questions. Okay, I'm going to take that as a no. Just antibodies are defensive proteins carried in the bloodstream. Figure 3.2 shows a simplified, incomplete structure of an antibody. Complete the figure by drawing and labeling bold letters. You must do both, and we've only got 1 mark to get here, so not worth taking huge amounts of time on it. The missing parts of the antibody. Okay, so we've got to do that and label it. What are we missing here? Do antibodies have a tertiary or a quaternary structure? You can answer that just from what I've given you here on the diagram tells us that. Okay, so this is a polypeptide chain, and this is a polypeptide chain, plus it's missing the chains on the outside here and here. So there's multiple polypeptide chains. Therefore, we'd say it has a quaternary structure. I mean, you could say that the quaternary structure includes a tertiary structure, but they would be looking for, if it's got more than 1 polypeptide chain and you had to choose between tertiary and quaternary, you would say quaternary. And they are joined by these cross-linking bonds. Okay, so the structure is this bit, these tips here. What do we call these tips on the end here? Antigen binding site or the variable region. Okay, so minimum of 1 light chain drawn on the outside of the heavy chain. So this bit here that is drawn up to this line is the heavy chain, and then the light chain goes on the outside. Very few questions actually determining the difference between that. And a label to the light chain, light polypeptide, or this means slash means or the variable region, or the antigen binding site. And this doesn't say, so you have to draw the chain and you have to label one of these things. Okay, I wonder if there's more on that. That's just a simple one that was just I didn't do any content on that. So there's a bunch of there's another one here, but I've only got it with the mark scheme, so I'll share it with you and we'll go through it. Okay, so another molecule that relies on specific shape. The previous question was about enzymes. Bind to a specific compound is an antibody. This shows the generalized structure of an antibody. And again, we've got the similar kind of thing, but we haven't got it's a different way of, it's a diagram, it's a different way of representing it. I was mentioned the hinge proteins yesterday. You can see those represented here. So we've got this Y being there. So draw a ring to show the part of the molecule that has a shape complementary to that of the antigen. Well, they've got these like triangular, these are the variable regions or the antigen binding site on the end here. So you can see like this is acceptable, and this is acceptable. So the component Y on the antigen is a bond. State what type of bond this is and give its function. So what kind of bond is it? Well, this is a real physical, it's a covalent bond, and that we call it a disulfide bond. So this is between two cysteine amino acids and it's got to be like a physical hardcore bond because these chains are, otherwise they're not attached to each other. So it is a disulfide bond and it holds the heavy chain to the light chain. These are my documents, so no, you don't have access to these. But you've got access to all the past papers and all of the mark schemes, but not this specific document. So these are the kind of questions you get on antibody. I might switch exam boards now and do find another one on would covalent bond still be? Covalent bond is a very broad class of bonds. So if I would say the only covalent bond you need to know in A-level biology is disulfide. So I would just go with disulfide as the one to learn. You can spell it either with PH or with an F. That isn't important. Yeah, I would go with, I don't know whether they would accept covalent. It's such a broad term that I think you run the risk of not getting the mark if you say that. I mean, obviously it's the right class of bond, but well, am I back at question 1 already? Just finding this question here. Question 6 is going on forever. Oh, okay. Whooping cough is a disease that is particularly serious in young children. Whooping cough is caused by a bacterium, Bordetella. I don't know how you pronounce that. Pertussis. Children may be vaccinated against whooping cough, so you can be vaccinated against it. In an investigation, a group of rats was vaccinated. So it's not being done in humans, and obviously we're interested in humans. It says children here, and this is rats. So just in my head, that's something I'm just taking note of as I go through the question. 60 days later, these rats were infected with the bacterium. In this investigation, the levels of 2 antibodies in the blood were measured. So there's 2 different antibodies going on here. The graph shows the mean levels of antibody A and antibody B. So presumably this is antibody A here. So this is the one line. So the vaccination obviously contains antibody A. However, after infection with the actual bacterium, there was also an immune response to antibody B. The A2 papers are significantly harder than the AS papers, yes. Not only in terms of the content, but the skills that they examine. There's much more application in them. Okay, so that's how many antigens did the bacteria possess that cause an immune response? There are 2 because we've got antibody A and antibody B. I mean, there's probably the surface of that bacterium will have had way more than that, but there's two that we've got information on. For antibody A, compare is the command word, the increase in mean level after the vaccination with the increase in mean level after infection. So mean level of antibody. It's a bit weird they didn't put antibody in the question here. We've got 2 marks to get. So what can we say? 2 marking points. Well, obviously we can talk about the peak. What else might we want to talk about? This is basically describe how secondary response is different to primary response, but obviously we've got to use the graph to get the information. Okay, so we say the mean level of antibody in arbitrary units is greater. Maybe give a value. Here it's going to be 25. Here it's going to be, what is that? I can't see it. 13, 12, I think it is, 12. What else? Shorter lag time. Okay, so the duration between the here and the peak, and the duration between here and the peak. It doesn't look that different, but I imagine it will be shorter. You could do compare the increase. A percentage increase isn't going to work because you have none here. So any 0 to 0.001 is like an infinite increase because you're starting at 0. So it's a nice idea to do a percentage increase. Oh, you mean a percentage increase between the peak number here and the peak number here? You could do. I was thinking you can do a percentage increase between this and this versus this and this. You could talk about the rate potentially. So how many arbitrary units of antibodies are produced per day would be one thing that you could compare. Okay, what I might do, I can actually be a bit clever here. I think, okay, I'm going to set you up with the next question. I'm going to get that mark scheme in a different application, and then I'll tab between them. Here we've got to explain the changes in mean level of antibody A after infection, so we can ignore antibody B. Which I'm assuming that you're talking about the previous question here. The command word, we only ever obey the command word. We are comparing and doing comparisons between the two. There would be no marks for explaining, especially if you had explained it here, and then you get to this question, which says, explain the changes. Then you probably, it's like, oh, I've already done that, but that's probably not going to get me marks for that previous question. It's actually those marks are going to live in this question. So this is a 3-mark question on the reasons why there's a mean change in antibody A after infection with the bacterium. So this is after the actual infection. I'm going to leave my screen share paused on that screen. I'm going to go and find Yeah, I have to unpause my. So what's the question? Now that I've put the graph back on the screen, you've got to make sure the question's clear in your mind. But it's really important. This is a good exam skill to practice. Having that, if you now don't know what the question is, my question to you is, how well were you going to do answering that question? Writing an answer to a question that you didn't really have in your head. It's really important you have the sort of key parts of that question in your head so that you can answer it well. You know what the command word is, you know what that question is asking you to do, because if you don't have that in your mind, it's going to be very difficult to answer it well. Jacqueline, the previous command word was compare. So it's like this versus this. This is this, and this one is this, and this is how they're different. Or this is this, and this is this, and that's how they're the same. Typically in the new spec, you'll see compare and contrast, which means give the similarities and give the differences, but not reasons behind them. So what, again, the question was, explain or give reasons for the change in antibodies after infection with the actual bacterium. So we can ignore the peak after vaccination. It's the secondary peak for antibody A, not for antibody B. Okay, memory cells present, shorter lag time. So those, you could maybe say the memory cells divide by clonal expansion. They make and secrete many antibodies and release them into the bloodstream. Does it tell us here it is in the blood, right? So probably worth the secretion part is important here because the antibodies are in the blood. So that's where it would possibly be a good time to differentiate between the production of antibodies and the secretion of antibodies. That how they get into the bloodstream is when they're secreted. Okay, we don't have much time left. I've got to go get ready for my year 13 session. I'm going to go, I think. So the first question was compare the peaks. The second question was explain the second peak. If it's going to find it for me. It should have taken me straight to the mark scheme, but okay, so the compare question. Levels of antibodies rise soon after infection. Levels of antibodies rise faster, so sooner and faster after the infection compared to the vaccination. Levels of antibody rise higher after the infection. So less lag time, steeper gradient on the graph, higher peak on the graph. Each of those three things. And this needs to be a comparative, ER. It's faster. You can't say it's fast. You must say it's faster than the vaccination peak. So the ER is important, except converse arguments, you can say it's slower, etc. And a comparative manipulation of data. It's this many times more. So you could probably have done a percentage increase between the two peaks after the, but you'd have to do it the same number of days after the peak. You need to be a like for like comparison. And then the reasons, well, there was no mention of secondary immune response. So whenever a key term is missing from the question, you really want to make sure you're including that key term in your answer because it's just asking for a mark. Reference to memory cells. Yeah. The idea that on secondary exposure, memory cells are activated, cloned, or stimulated. You could say memory B cells differentiate into plasma cells would be acceptable for that marking point. That secondary response antibodies are released from the plasma cells. So secreted would also be accepted there. Check out the schedule in the Exambulance. If you don't know what the year 13 session is on, I can't remember off the top of my head. That's why I'm telling you to do that. But go log into the study at home Exambulance and you check it out there. The lag time is the delay between the vaccination or the infection and the first production of antibodies being produced. Correct. So compare and contrast. Comparison is looking for the similarities. Contrasting, it would be looking for the differences. If it just says, it just says contrast. This is an old spec paper, so they would generally are going to say compare and contrast in the new spec, and typically they're 4-mark questions rather than 2-mark questions. This one, it said compare. So you're basically saying, how are they different? If it just says compare, then I would looking at how are they different more than how are they the same. If you've got nothing else to put and you have got something that is the same, then we can state similarities as well. But I would go differences first and similarities after if it just says compare. Okay, sweet. I am going to go and get my notes ready and charge my iPad for a few minutes because otherwise I'm going to be out of juice. See you. Okay, so next week, inside the Exambulance, you will need to use Zoom. There will be a timetable updated over the weekend so that you can easily join us inside those sessions. We might do a little bit on YouTube. I'll be doing more on YouTube in the evenings, but less teaching, more other stuff. Let me go find the control panel and say goodbye for now.